So you've made it to another lecture module. Good for you. Look how well you're doing this semester. And this lecture module is going to concern DNA. So a large part of a forensics laboratory, it's blood alcohol content. We're testing that almost every single day. It's a large majority of the cases, as well as other toxicology tests that we might be doing. But another large test that is commonly done in a crime lab is going to be DNA. All right, so we're going to talk about what DNA stands for and what it represents. So long ago, DNA was often called the stupid tetra means four, nucleotide. All right, and the reason they said this is because they thought that DNA was just a random assortment of molecules clumped together that had no importance at all. When we today think about our genetic carrier, we often think of DNA. But this was not the case in the very early parts of this discovery. Back in the day, they actually thought the genetic carrier were proteins. And the reason is because there were so many different proteins that were out there, so many different choices, and everyone had different proteins maybe, and that is what made them different from each other. If you take a look at protein structure and the way proteins are made, they are made from 20 amino acids. And these amino acids get hooked up in different orders. And those orders of how they basically are chained together gives rise to different shapes of proteins, different functions of proteins, and everything else in between. The problem is that DNA has four. So early researchers looked at these two options. And they said a DNA molecule has only four options that we call nucleotides or bases. And they can come to us in a different order. That's not a big deal. We know that. But compared to proteins that have 20 choices that we could pick from, it looks like if we are that drastically different, because we do not look the same, act the same, think the same, move the same, then maybe proteins is going to give rise to that differentiation that we can see between each other. Well, long story short, those researchers that thought that proteins back in the day were the genetic carriers were wrong. And this stupid tetranucleotide that they thought was just a waste of a molecule was actually the genetic carrier. So how did all of this kind of get started? Back in the day, well, we have a doctor, and I'm going to call him Mr. Puss. He was Swiss, and he wasn't a chemist. He was actually a physician, a doctor. But he understood the importance of organic chemist, and he began working as an organic chemist in the late 1800s. So we're talking here about 1860-ish plus. So one of the things that happened here is that as a doctor, he practiced, but he came down with a horrible case of typhus, and he became partially deaf. And he claimed that he was a disadvantaged doctor. He couldn't hardly hear people or his patients. And he knew that there was an issue, and he knew that he had to change careers. So as a doctor and as an orgochemist, he blended the two, and he was very concerned with leukocytes. Oh, where we've seen that word before. Yeah, in our blood lectures. And leukocytes, of course, were the white blood cells. There was something special about these white blood cells. We needed to know more about them. They were very difficult to obtain, though. So what did he do? Well, like any good scientist, he kind of came up with a very creative way to get what he needed. And he would go around to the hospitals, and he would go around to people that were suffering an injury or some type of illness, and he would collect the pus, because that was red blood cell or white blood cell containing. He would collect the pus bandages. Kind of disgusting, right? I mean, could you imagine this guy going around every other day, going to these hospitals and saying, "Could I have your pussy bandages from your patients?" But this is exactly what he did. So all of these bandages were brought back inside of the lab. And I'm going to put a quote here around lab, because was it really a lab like we're thinking about today? No, not really. And the question is, how could we get the pus off of the bandages so that way we can test them a little bit later? Well, what he discovered is that he could extract the pus, and in particular the white blood cells, by using salt. And this salt was a solution of sodium sulfate. So he would soak these bandages down into salt water or a salt solution. And what happened is, this is my vat, and here is my solution, and let's say that this is my bandage. It's soaking, it's hanging out. And over a course of time, a gummy substance fell down here into the bottom. Now, there were no centrifuges during this time, right? So keep that in mind. This could have been sped up a little bit quicker if we did have some, but no high-powered centrifuges. So this stuff here in the bottom, he gave a name, and he called this nuclein. And a star was born. This substance was slightly acidic. So because of that, we have a name change. You know, kind of like Puff Daddy or Prince. And this name change went from nuclein to nucleic acid. All right, well, what was Mr. Puss's name, right? He was kind of the first one to give credit to the extraction of nucleic acid, so that way we could do further studies on this type of material. And this was a macromolecule, meaning that we could actually see this stuff because there was so much of it. Well, his name was Friedrich, Friedrich Miescher. And he is going to be given credit in the very beginning stages of nucleic acid and the studies that are going to happen afterward. All right, so this is the late 1860s. So then we're going to have someone else that comes around after Miescher. So let me clear this off. And now we're talking about after 1860. And this man's name was Oswald Avery. He had a famous quote. He said, "It's fun to blow bubbles, but you need to prick them yourself before someone else does." All right, so be creative, have fun, but make sure that you take the credit for it. Don't let somebody else ruin a good thing that you've got going on. Oswald Avery was Canadian, but he moved to the United States. He became an American physician, and again we are seeing another physician. We're not really seeing a chemist. And he was a very main, large personality when it came to immunochemistry. That's right. This is a branch of chemistry that deals with your immune system. And in 1944, he's going to come around and he's going to say, hey, you know what? DNA is the material in which genes and chromosomes are made. He never received the Nobel Prize. Never. But look at the statement that he made. He did not say that DNA was the genetic carrier. What he said is that DNA is the material of which genes and chromosomes are made. Genes and chromosomes are the genetic carriers, not DNA. Well, we kind of know that's not really the case now, right? We know that all of this is based on the DNA and the nucleic acids that we're getting ready to talk about. So how did he kind of discover this? What was going on during that time? Well, Oswald Avery basically did a series of studies. Let me pull up a picture. And this is what he did. So he goes into a laboratory and he takes what we call a rough strain. And a rough strain is basically just maybe some bacteria or a virus. And it wasn't alive. They were dead. So he injects this dead virus or bacteria into a rat, and the rat lives. Yay! Well, this was not surprising. We knew that this was going to happen. No big deal. And then they took a smooth strain, and a smooth strain was basically an alive version. And they took this live creature, if you want to think of it that way, that's kind of what they are. Kind of weird. Grosses you out when you see them under a microscope. And they injected that into a mouse. Well, it was alive. We knew that this mouse would probably get some type of disease. And sure enough, the mouse dies. And then we take heat-killed smooth strain, which means these were alive, and we basically slocked it full of heat, thinking that we would have killed the virus, and we shoot it into a mouse, and yay! That mouse lives. So this shows us that we could kill viruses and still put a dead virus inside of your body, and your body would be okay with that, and you would not have any symptoms at all. You'd just go about your daily business. And then finally what happened is that they took a mix, a rough strain, and a heat-killed smooth strain. And they said that, this one's not going to be a big deal. I mean, this one is rough strain, which was dead, and this one was heat-killed, and that we regarded as dead. And if we go back to original studies, well, this rough strain, the mouse lived, and the heat killed, the mouse lived. No big deals there. So if this one lived and this one lived, then we can mix these two things together. We'll shoot it into a mouse, and it will, that mouse died. Wait a minute here. We had two dead viruses. One was dead. The other one, or non-virulent, not quite dead, just didn't carry a virus. And then the other one was heat-killed, which did carry the virus. Oh, so that's what that means. So here, it's not really dead, right? That just means that this is not carrying a virus. And because it's not carrying the virus, it's not going to give it to the host. But they're still alive. Now there's the difference. Because this one was a smooth strain that carried a virus. and we killed it. And those dead little bodies were mixed in with these things that were still alive. They just didn't carry a virus. And we mixed these two things together, and they had a party, and they made out. And it killed the mouse. What is this trying to show us? Well, something is transferring here. If this did not cause the mouse to die, the original ones of these caused the mouse to die, but we heat killed these, so they should live. Something is happening between these two things, transferring in a way this non-virus strain over to a virus strain. This is going to give rise to the first concept of transfer and what is exactly causing this type of thing to happen. So guys, here's Mr. Puss. This is the man that would go around and collect those bandages for you back in the day. And then if you want to take a look at Oswald Avery, here's Mr. Avery. So in the next video, what we'll do is we'll talk about the research and the follow-up to all of these studies that these two men did and donated to science, basically. I mean, they were first initiation of DNA and genetic material, and it was all happening right here in the late 1800s and early 1900s is when this began to get started. So in the next video, we'll come back. We'll talk about some modifications that built upon Avery's studies, but was really kind of initiated by Mr. Puss back in the day.